Section 6.5, traits and probability. For this video, you're going to be taking notes on notebook paper. Be as exacting as you can and make sure you're getting all the info necessary. First thing we're going to talk about is it's called a Punnett square. Okay, this is a Punnett square over here. Okay, and it's a diagram showing the different types of gene combinations that can result from a genetic cross. So in this case, we're getting a cross of one parent that is heterozygous for this trait designated by letter A, and the other parent is also heterozygous. Okay, and it shows the possible gametes that form, and these are the possible outcomes. So on the top here, that is the gametes that can come from one of the parents, and on the side, these are the gametes that can come from the other parents. And you cross these guys, columns times rows to get the genotype of the offspring. It doesn't matter which parent you put on the horizontal side or the vertical side, okay? But for just in terms of staying consistent, I'm always going to write it as the first one listed is going to go in the horizontal spot, and the second genotype listed is going to go in the vertical spot. Okay, so in this case, this is saying that this is the father, right? Parent's father. So dad can give a dominant uppercase A or a recessive lowercase a in its gametes. Mom can do the same thing, either a dominant A or a lowercase a. When we put these guys together here, column times row, we get homozygous dominant. This one would be dominant and recessive, so we get a heterozygote here. Column times row, we get another heterozygote, and this one would be column row, we get homozygous recessive. Okay, so another one practicing here. This one, this time the trait is designated by the letter T. So we have a dominant T and a recessive T coming from one parent, and again a dominant T and a recessive T coming from the other parent. Okay, the way you do this is column times row, and you write the genotype of the offspring in there, which is TT. For this one, column times row, and we get capital T, lowercase t. When you're listening to genotypes, you're always going to put the uppercase letter first. Again, this one is going to give us capital T, lowercase t, and this one will give us lowercase t, lowercase t. So genotypes are 1 homozygous dominant, 1, 2 heterozygotes, and 1 homozygous recessive. Okay, so for this one, you're going to get this cross, homozygous dominant, big T, times homozygous recessive, little t. Okay, to set up our Punnett square, we're going to put one parent on the top, and like I said, I'm always going to do the first one across the top. So the gametes that could form from this homozygous dominant would be big T and big T. Those are the gametes that would form from that parent. This guy would be the second one. This would be little t, right? So we have the two recessive alleles from the second parent. Okay, and again, columns times rows, we're going to get big T, little t. This guy, big T, little t, vertical column times horizontal row. Okay, big T, little t, big T, little t. So in this case, all of the offspring are heterozygous. So 100% heterozygous, right? Heterozygous, okay? There's no homozygous dominant because the one parent was homozygous dominant, but none of the offspring are. There's no homozygous recessive because this other parent was homozygous recessive, but none of the offspring are. So when we're looking for the genotype and phenotype of the offspring, we're getting 100% heterozygous, and these would all take on the dominant trait, which would be uppercase T. In this case, if we're dealing with Mendel stuff, it would be for a tall plant. Okay, so let's do another example here. This one, we're going to have one parent that is heterozygous for a trait, and the other parent that is homozygous recessive for the trait. So again, to set up our Punnett square, I'm going to take the first parent, and I'm going to put the gametes that would come from their genotype, which would be 1 capital H or a lowercase h. Those would be the only possibilities of them getting, or that would go into their gametes. The second guy, this homozygous recessive, would be little h and little h. We do our cross, right? Columns by rows, H, H, filling in the grid, little h, big H, little h, and little h, giving us a genotypic ratio of 0 homozygous dominant to 2 heterozygotes to 2 homozygous recessive. So we would get half of our offspring showing the dominant trait. The heterozygotes would show the dominant trait, and a half of the offspring showing the recessive trait, this whatever the trait would be. Okay, another cross we have. This is heterozygote and a heterozygote. So we would put one parent along the top, which would be this guy, big T and little t, because a heterozygous parent can give either a dominant allele or a recessive allele to the gametes. Okay, and the other parent, this one would go on this side. Again, one dominant or one recessive allele because that's what's in their genotype. When we do the crosses, we would have homozygous dominant, big T, and a heterozygote. And this one would give also a heterozygote, and this one would be a homozygous recessive. So our genotypic ratio would be 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. Okay, and the phenotypic ratio would be 3 talls, because this has the tall gene, so 3 of those would be tall, and this guy would be short. All the Punnett squares we've done so far have been dealing with just one trait with 2 different versions of the allele. These are called a monohybrid cross. Okay, so we have one trait given designated by the letter A, capital A being the dominant version of it, and lowercase a being the recessive. Okay, so one trait with 2 contrasting versions of the trait is called a monohybrid cross. We can use Punnett squares to predict those. So Punnett squares can be used to predict more than one trait and how they're connected to another as well. All the ones we've done so far, we're dealing with just a mono or just one trait, but this one demonstrates a dihybrid cross. Dihybrid cross meaning there are 2 traits that have 2 different versions of the trait. Okay, so here we have homozygous recessive for both the seed color and the seed shape, and this is homozygous dominant for both the seed color and the seed shape. So big R is round, and the other version, little r, would be wrinkled, and big Y would be yellow, and little y would be green. So these are the 2 traits, right? Seed color and seed shape, and they're alternating versions of it. Mendel used this idea to come up with something called the law of independent assortment, which says that the alleles for these different traits separate independently during gamete formation, meaning that A, they're not on the same chromosome, and that different traits, because of this, are inherited separately. So what if we have a phenotype, dominant phenotype, but you don't know what the genotype is? You can do something called a test cross, which would allow you to figure out whether or not it was homozygous dominant, displaying the dominant trait, or heterozygous, it's heterozygote playing the dominant trait. So if it's showing the dominant phenotype, it could be either, and this is in this case for purple flowers, it could be homozygous dominant big P for purple flowers, or a heterozygote big P, little p, which would also show purple flowers. In order to do that, you run something called a test cross, where you cross the flower displaying the dominant phenotype with a homozygous recessive flower. Okay, in each case, if, in this case, this one was big P, so that's the gametes for this big P, they're all going to be heterozygote, and they're all going to be purple. If it was big P, little p, that you're crossing with this guy, you would get this version, right? Either big P or little p, and we would get half purple and half white. If you're getting any of the recessive traits showing up in that test cross, or after that test cross, then you know that the genotype of that flower or whatever organism is displaying that dominant trait you know that it was heterozygous, because that would be the only way that it could come up as recessive in that generation. Okay, I mentioned this a little bit earlier, but I didn't really explain it as well as you might need to. Punnett squares are also used to determine the ratios of traits. Now, if we're talking about just the physical look of the trait, that would be the phenotypic ratio. Think of it like the physical traits that are being shown. The types of physical traits being shown is the phenotypic ratio. The genotypic ratio are the alleles that that particular offspring would have inherited from its parents. So in this example, where we have a heterozygous parent, right, being crossed with a heterozygous parent for tall flowers, we get this one. This is homozygous dominant, right? Then we get 2 heterozygotes, and we get 1 homozygous recessive. So the phenotypic ratio for this is 3 tall, because that would be the dominant trait, to 1 short. And the genotypic ratio would be 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. Again, Punnett squares can be used to predict not just the phenotypic ratios, but also the genotypic ratios, what the organism would look like and what genes they would have inherited from their parents. You can also use Punnett squares to calculate the probability of inheriting a particular trait. Probability deals with the chance that a given event will occur. Okay, so for example, if you're flipping a coin, the probability of you getting heads when you flip the coin would be 1 out of 2, because you'd either get a heads or a tail. So there's 2 possible outcomes, heads and tails, but you're only going to get one of those when you flip it. Okay, so the probability of getting heads if you flip a coin would be 1 out of 2, or 50%, or 0.5. That's what you could represent it, right? In this case, what would be the probability of getting a homozygous dominant? Well, if we have 1 homozygous dominant and 2 heterozygotes and 1 homozygous recessive, the total number of outcomes is 4. Okay, so if we're only getting 1 homozygous dominant out of this 4, the probability of getting homozygous dominant is 1 out of 4, or 25%, or 0.25. You could express it any of these ways. The probability of getting a heterozygote would be 2 out of 4. 2 out of 4. Again, 4 possible outcomes, and 2 of these are heterozygous, which would be 50%, or 0.5. Probability of getting a homozygous recessive would be 1 out of 4, again, 25%, or 0.25. All of these possible outcomes have to add up to 1 or 100%. They have to. Okay, this last problem is describing something that we're going to have to. This would be like a word problem you'd use for genetics. Okay, it says you're getting a heterozygous cross, and we're giving it the traits. So capital T would be tall, lowercase t would be short. So in a heterozygous cross, both parents are heterozygous. So what this cross would be big T, little t, times big T, little t. We've done this one before, so it should be easy. Then it asks us to predict the genotypic ratio and the phenotypic ratio and the probability of having a short offspring. So use the Punnett square. Okay, the parents would give either a big T or little t from the first one, and the same big T and a little t from the second one. When we fill in our Punnett square, this is homozygous dominant, this is heterozygote, heterozygote, homozygous recessive. So the genotypic ratio would be 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. The phenotypic ratio would be 3 tall to 1 short. And the probability of having a short offspring would be 1 out of 4, or 25% could be short.